And I think, you're right that having that side, whether it's art or music or, having a digital landscape where you're being creative with or writing, whatever you want to do outside of science, I think it does help creativity in a sense too. I would 100% agree. I have no proof of that, but I would agree that it does help.
That's your science side talking.
Exactly.
You're looking for proof.
Yeah, exactly.
Hello, everyone. My name is Jeremy Fortner. I'm the active president of San Diego Computer Science. San Diego Computer Science is a tech incubator for college students founded in San Diego. Today, I have the pleasure of speaking with Dr. Afshin Beheshti. Dr. Beheshti is a renowned researcher in the field of space biology, bioinformatics, COVID research, and many other things. So without further ado, I'd like to introduce Dr. Beheshti.
Oh, thank you. Thank you for the introduction. Thanks for having me. Excited to see what we talk about.
Yeah, so I have a bunch of different types of questions that I feel like I would really be interested in having your feedback on. But first, I'd like to just go through and ask you kind of what inspired you to get into research, what inspired you to go into biophysics, and it seemed to have all kind of added up with where you're at now at NASA, but what inspired you to start a research career?
Yeah, I mean, I've had a unique, as you probably saw, unique path to get to where I am. But yeah, no, I guess what inspired me to do research was basically, back when you're kids in high school and before that, science was always fascinating for me. So, as a kid, I had one of those little toy microscopes. I would put little slides on, put a little water or insects underneath, and look at that as a kid. So that was fascinating as a kid. I was like, okay, this is kind of neat, the whole world around you in science. And then eventually, when I was in high school, I started getting interested in physics mainly. So I learned about physics, and then once I was doing it through undergrads, I was doing more high energy physics. So that was my path to, which is completely different from what I'm doing now, but it's somewhat related because the physics is a basic understanding of the world. But then eventually during graduate school, I was gearing more towards the biology field, but it's more biophysics. Understanding how, say, physics might be applied to. I was looking at how DNA, for example, might move or through objects. I didn't care about the biology aspect of it. And then eventually I switched over to biology after my PhD. Did a postdoc in an oral microbiology lab, and then cancer systems biology, and then got involved with people who were doing, I had NASA grants, so NASA research postdoc I was doing another lab. But yeah, so I mean, the inspiration basically was curiosity, I think, was the key. So I think a lot of scientists like yourself, or budding scientists, I should say, that was because most other fields, curiosity is good in other fields, but it's not as rewarding as you do in science.
Cool. So tell me a little bit about the work you're currently doing at NASA Ames.
Yeah, so at NASA Ames, I do several different projects there. Initially, when I first started, now about 6 years ago, I joined to work on the Gene Lab project, which Gene Lab is this open science, open platform, tool and resource to the community where it allows for researchers to dive into omics data, basically big data, right? Like when you do the sequencing in RNA or do protein analysis, like big omics data, as it's called. So all that data that people have done, and various investigators, it gets dumped on this platform that now has been developed into this very nice resource that anyone in the community, anyone in the world can dive down to this data and use it. And you don't have to necessarily have a computational background. If you do, that helps. But it's been designed in a way that people actually, without any computational background, can go in and search like, oh, I've heard this gene is my favorite gene or favorite protein. How might this change in breast cancer or kidneys or other tissues? And then you might just do a search and you can have a hypothesis generated. So that's what I was initially working on. Now the platform is now being developed even more. But in the past couple of years, in the process, I end up getting my own grants, as you do in academics and government. You end up getting you could apply for research grants, and then you get grants, of course, and then well, not of course, but eventually you would hope to get grants. It's a long, that's a whole other question we could talk about and answer. But eventually got my own grants of some of my own research and ideas. So some of my own ideas and interests are relating with small RNAs, which is, there's, in the classic sense of biology, there's DNA, right? And then DNA transcribes and makes the RNA, and then RNA can translate into proteins. But in the past couple of years, in the past couple of decades, 20 years, people discovered these really small RNA called non-coding RNAs, and one aspect of them are microRNAs. So these RNA don't actually translate to protein, but they're very small. So people 20 years ago thought it was debris and threw them out, garbage. But some smart folks, again, curiosity aspect, looked at these and they're like, oh, these are actually very functional and control a lot of things. They don't make proteins, but they don't translate into proteins, but they actually regulate hundreds and hundreds of genes and can block protein translation from regular RNA. So these things are involved in their healthy aspects floating around in your body, and also things related to disease. So I've been working on these microRNAs because my hypothesis is that these microRNAs can actually be, this would be a signature of these microRNAs related for every kind of disease type. So say cancer would have its own signature related. Spaceflight should have its own signature microRNAs circulating in your body related with health risks associated that, whether it's cardiovascular risk, renal risk, kidney issues or liver. There should be a signature that might be universal, and there's components of that related to different types of health risks or cancer and so on. So that's where I got grants to look at that, and been pretty getting some papers and successful at getting nice results to show these microRNAs might be key regulators in space. Another aspect I work on is like mitochondrial. Mitochondrial is probably, you hear the classic thing, this powerhouse of the cell. Of course, it's a lot more complicated than that, but the simplistic view, it's a powerhouse of the cell. But that's another aspect where we collaborators have been working on a bunch of aspects related to mitochondrial might be a central driver in space flight response or health risk to your body. And then there's a bunch of other projects within the space biology context I work on, but I don't sit still. So I also have things that I work on cancer specifically, not related to space, but it could be connected. And then COVID, when COVID first started, I formed this COVID group, as you mentioned, I work on COVID. So, there's a lot of projects outside of that, outside of the space biology world, that we work on COVID research. There's some grants associated that we've received that, that then me and my collaborators that we've been working on COVID work. So a lot of this stuff, of course, you need the money. You can work on this stuff on your own, off the clock, which we do a lot. But then, eventually you need money to fuel the research too. That's when you apply for the grants. So there's grants we have now for different COVID research. And then there's a bunch of other little side projects that we have going on, or I have going on, that related with different things that we could go into. But in a nutshell, that's the long answer to your question.
So you were mentioning microRNA and mitochondria as being the main sources of being able to indicate like biomarkers when it comes to different body systems and how they work within space, like muscle tissue and skeleton. And so I guess I'm kind of speaking from a non-professional perspective, but microRNA and mitochondria, there's something that can indicate a multitude of different features going on within the body. It's not just one thing that it can tell. You can read a bunch of you can get a lot of data out of one system. Yeah,
so the microRNAs, they're two different mechanism systems, and they both can actually relate to each other. So microRNAs themselves, since as I said, they're very small, there's thousands of them around now, and a lot of these, since it's only 20 years old in the biology field, that's pretty much in the infancy, right? So there's still a lot of information that's not really understood about microRNAs. But what we do know, based on that, what they do is, since they regulate genes basically. So a microRNA, is a small single-stranded RNA that floats around in your body, and this also could be in your tissue expressed. And what happens is it's usually around 20 some nucleotides plus or minus, a few nucleotides here and there, depending on the microRNA. And there's a region on the microRNA called a seeding region, and that seeding region is what binds to your genes. So that's going to bind to like all the genes that you might not. So for example, in cancer, there's microRNAs associated with that. And there's tumor suppressor microRNAs, meaning those are good ones, right? So they bind to the oncogenes, so the cancer promoting genes, and they suppress it. So once they bind to it, they silence those genes. But then there's oncomeres or tumor promoting microRNAs that would bind to the tumor suppressor genes that, or bind to things that might then cause cancer to promote. So then the microRNA is associated with every aspect of disease related to that same kind of example. So the mechanism that a microRNA might work with is that's either binding to the either if it's a beneficial one, of course, binding to things that would cause a disease, or binding, or the the ones that associate with the disease will bind to the ones that would be healthy genes. So typically, let's say for spaceflight, you got radiation is one, space radiation is one aspect astronauts exposed to. Another part is microgravity, or the reduction of gravity, or virtually no gravity, right, in space. And that in itself would cause, let's say, the space radiation, would cause physical damage to your cells, like DNA damage, for example. And that DNA damage might cause things to happen where be like oxidative stress activity, or different kinds of pathways like immune suppression, or different immune related activity might happen to cause things to happen. So once that happens, then these microRNAs that might not be floating around in your body that are associated to negative things will get expressed. So this damage will, and then also will express good microRNAs to hopefully counter that stuff. So these microRNAs usually counter, the bad ones are usually expressed in the body due to some insult to injury, whether it's, cancer, or whether it's now space radiation or microgravity. Now the mitochondrial aspects is like, yeah, that's different. So, in your body, mitochondria is basically, there's there's their own organelle in your body, right? And some people think from evolutionary, they used to be a bacteria a long time ago, millions and millions, billions of years ago, used to be a bacteria. And evolutionary wise, they realized, oh, this is beneficial to have in your body. So it incorporated into your body. So you can think of it as a potential microbe long time ago that now is, established as mitochondria in your body. So the mitochondria itself, that's used for a lot of good things in your body. Of course, you need that. It's because, it's for cell respiration and to keep you active and going. You need that for calorie consumption, energy production. And as you age, mitochondrial functions decline. That's one of the natural processes of biology. So as you age, and then that's what happens when when you age, you have less energy, for example, and less ability to combat disease. And usually mitochondrial, well, depending on who you talk to, if you're really mitochondrial researcher, you consider a mitochondriac. That's what we call ourselves. Or a mitochondriac, then we'll say everything's mitochondria centric, or that's what typically happens when someone specializes. But in this case, we might be right. But in the case like mitochondria, sometimes you have dysfunction, then that affects your immune system. And then the immune system dysfunction will be cascading to different things. If your bioenergetics is affected your body and dysregulated, let's say with your space flight, that suppresses and overactivates different things in mitochondria, which is not good. That's going to affect your circadian rhythm and your sleep cycle. So we know that's affected. So there's a lot of things. Now, connecting microRNAs and mitochondria together, there's actually microRNAs that come from mitochondria, and those are called mitomeres. So this is where the mitochondria connection and microRNA link is. And just like I said about microRNAs and how everything's the downlink the process of it binding to genes and causing all the downstream effects of it doing that, it could happen the same thing. So with let's say if there's an insult to the microRNA of the mitochondria, let's say it's damaged from space radiation, for example, then it would probably release, as we know, it would release some or overexpress some microRNAs that would start binding to genes that could impact the mitochondria further, cause further dysfunction. And then that's the cascading effect. And then when it does that, then of course the mitochondria will start get accelerated further and further to cause dysfunction. And then then it's the downstream impact that their bioenergetics will be affected in space. That it could relate to some of the cognitive issues, for example, astronauts see, and other factors.
Interesting. So you mentioned NASA Gene Labs, which is a open source data repository. And so NASA will go and conduct experiments in space on the International Space Station. They'll and then send the data to that repository. Is that correct?
Sort of, no. So yeah, but it is, so there are experiments. So you typically what NASA Gene Lab is just a repository, right? So that's where any omics. So what happens is, let's say I have some NASA grants that I received, and various principal investigators would apply at universities. They get their own NASA grants to do research. And you don't have to be a NASA to get NASA grants. You can be anywhere, right, in the US to get funded. And you just have to apply for the regular grant systems that we all do in academics that a lot of people outside of academics are not familiar with, but it's a regular NIH grants people would apply for. NASA grants are the same thing. And it goes under a review process. And once you get a good score, then the program obviously look at it and then compare your grant to the rest of the grants to the good scores, and then hopefully you get funded. And it's a typically, it's a long process, but eventually once you get grants, it's rewarding. But so what happens is now in these grants, we budget money to run omics, and run, let's say, transcriptomics or proteomics or whatever experiments we do. We have omics in there that we want to produce data. And now, what is required, what NASA would do, since this is a government funded program, right? And I got a grant, so this is taxpayers' money. They said you should make this open source. This data should be open to the public once, which is good, open to the public once you published your first paper. You got a year, obviously to work on the data, and then so no one will scoop you because this is your idea. And then once you have this, then you put the data so the rest of the world can actually access this after a year. And NIH does the same for their cancer, like TCGA, the Cancer Genome Atlas. They have the same kind of concept. So any kind of government, I think, grants, they don't know that they should make this data available to the public, since it's public money being used to do this research. So that's what GeneLab is based on. So there is some data sets on there from actual internal money that they had to run sequencing on tissues from mice, for example, that was sent to space. That was sitting in the archive. So there's also experiments that payloads that from previous times where mice were sent to space from previous grants. Again, people applied for grants, and then they sent mice to space based on the grant they got to look at certain things. And of course, when they send the mice to space in the grant, they might look at like a specific organ, like liver, for example, or the brain. They're not going to look at the entire mice. So when these are precious, I guess cargo, you call them, or precious experiments. So they save everything, right? Since it's so expensive. And then all those tissue are archived. And then now NASA has their own ALSDA, which is the Ames Life Sciences Data Repository, which has the data from the actual tissues, but also have the tissues where you can make a request from, and then someone can run it. So within that, like NASA had done some sequencing, and GeneLab had that, and then others have requested money to sequence the tissue. And then so it's whatever, if you go on GeneLab, you'll see the investigator's name associated with that data set. So there's some data sets you see my name associated with that, since I had some grants around. You'll see data sets from other people in the community. So that's how it's built up now. There's like a bunch of data sets there. It's basically people in the community getting their grants. They run their omics. Now it has to be dumped on GeneLab. So it's kind of giving back to the system, right? And giving back to the world. But hopefully, that people then, because when someone works on a grant, they're focused on their study. And of course, there's thousands and many different ideas that you could get creatively work on these data sets that, that one person might not have thought of based on the same data that's published.
What advice would you have to students looking to pursue a similar career path to what you're doing currently?
Well, I mean, my advice basically would be do something that you won't regret working on. Basically, you hear most people go to their job and hate it, right? I love my job, so when I work, it's not work, it's fun, it's play. As my wife would say, it's like you just go and play all day. It's like, yeah, it's good, I enjoy my work, and it's fun being creative. And then that's the first advice I'll give students, is that you should not do it for the money. Of course, money helps, but not do it just for the money. But you should do it something that's gonna be rewarding for you, because eventually, you would find a career if you're doing it based on what you're really passionate about. Let's say in science, then the career would move along, because you'd be passionate of doing it. Obviously, you'd be good at it. Obviously, people will see how good you're working, and then you'll always have a job in science in that sense of it. Whether it's going in an academic route, whether an academic group is a whole different group than let's say going to industry. But the academic route could take a while to get established, because you have to apply for grants and move on. I mean, that's one set of advice. The other set of advice is that I was given a long time ago is that, and some people might not agree with this, but I would say don't be pigeonholed with just one idea, because as you see, I work on many different things, and it makes them more versatile. So a lot of the, let's say for computational folks, you have tools and skills available to actually work on many different subjects, from cancer to cardiovascular disease to cognitive issues, to CNS issues, central nervous systems issues. So you could apply that stuff to a lot of different things. And then you could specialize, for example, on one subject to really pin down what you want to do, or one, let's say, favorite pathway or so on. But if you just focus on one thing, that could really limit your career path into somewhere that eventually you're stuck, as some people might be, right? Or they think they are. But if you actually open up to say, hey, I could actually apply my ideas, and for example, this microRNA mitochondrial idea is now a plug and play for me. I could take this and plug it into the cardiovascular. I can plug this into COVID, which we've done. I plug this into cancer. It's just that, basically your education teaches you how to read and think for yourself. And then, you could read about, for example, if you started on the cancer field and you did this, and now, like, how do I apply this to space? All you have to do is read the literature, and then that's what your education teaches you after that, after a while, And if you're sticking in biology, a lot of things that you learn is wrong 10 years later, because that's how biology goes, right? The fundamentals are fine. Even the fundamentals change sometimes, but a lot of the things you're learning in school might change by the time, you're actually deep in it. And then you'll say, oh, okay. And that's how biology is, especially, let's say, if you're working on subject like I am with microRNAs. That's a very fluid field and then moving. And there's a lot of fields like that right now, not microRNAs too, but other biological features that people could focus on, there's not much known about, but are starting to be discovered. And the other advice is you should really, if don't just stick to wet lab. People should really start learning some computational tools to, like, the informatics side of it, because even if it's a little bit of R coding and just getting a little bit of it, it really helps. Because as we see, without omics these days, your field of research and science gets limited, and then you would have to rely on collaborators, which is good. You need a large network and a lot of collaborations to do science, which is another advice I'll give: make your network as large as possible. But having that skill and ability to actually do the computational part and interpretation of biology would be key to really make you a well-rounded scientist.
So the versatility of the different things that you go into kind of end up making you better at all of them, because you're not so stuck into one way where you can't go and make a pivot because you have all these different things. I definitely agree with that, because there's a bunch of different things that I'm involved in.
Yeah, and eventually too, you might get bored in the field you started out studying, like 10 years later. So you might as well have some way to.
That is true. So yeah, you mentioned getting skills with informatics. How have you seen, because I would assume that you've done research before AI and informatics were as prominent. How have you seen that the field of research has changed over time to where it is using AI more? It is using, in the field of biology, it is using bioinformatics. How has the field changed?
Yeah, when I first started, there's no such thing as bioinformatics. I don't think that terminology, I mean, it was there, but it was completely different terminology and definition. But yeah, so I mean, I had a physics background, so I had some coding experience. And when I first started, it was the microarrays were like the hot thing. It was like the chip, where they had the beads on there and probes on the bead. They had specific genes, and then you get different beads from different numbers of genes you want to look at. And one thing that's changed over the past 20 years, or probably 20 plus years, where omics and bioinformatics have evolved, is one thing's cheaper, obviously, to do it. When we first started, it was extremely expensive, and now it's so cheap that a lot of people just run it, and it's easy, a lot more used. The other part is, the bioinformatic tools that are there. I mean, back when I started, me and my colleagues would teach ourselves. So we'd be like, okay, how do we analyze this data? What's the best way to have the statistics to analyze this data? But kind of just at that point, early on, it was more discovery, figuring out how to best work with this big data sets and best be able to get this optimal statistics to do it. That's where, let's say, the adjusted p-value came from. P-value, for instance, moved on. And I mean, the most, I think, the most easiest thing that makes it easy right now, that's evolved for informatics to be more versatile and easier to learn than when I started, is that all the resources that's up there, right? So there's a bunch of packages, R packages and R codes that are there that you can look up. But if you don't know, what you do is you go on like this nice, very nice communities like BioStar, Stack Overflow, and you say, oh, you just type Google, how do I do this? And then you're going to find a bunch of forums out there. And even if like this, I didn't know R coding and I had to teach myself, that's how I started doing it. There's nice tutorials, but usually I'll be impatient to read the whole tutorial. So I'll just type Google, how do I do this? And then go in the forums and then be like, okay, I see this code. And then if I'm a newbie, when I first started, it's like, hey, I'm a newbie, I'm not familiar coding. And there's a lot of there's a lot of nice patient people in the community that would actually want to be there to help, and they get back to you right away. So that's the key. Like even for students that you don't have any coding background, the key is just to say, how do I, let's say, program to make heat maps or something, with informatics tools, and you could most likely just see a script, take the script, and then modify it, and then start playing around. There's a learning curve once you get over that hump. That's the key. So the biggest thing I think is these communities that are out there, and even now with these AI tools like Chat GPT and stuff, you can actually type in, how do I do this code, and it spits it out for you. I mean, the other aspect on research alone, AI and ML, early 90s and 2000s, there was AI and ML around, but obviously to run something, the computational power took much longer to do. So that my colleagues, like I have a lot of collaborators who do ML AI techniques to come up with scientific, let's say, for discovering how to repurpose drugs if you have genes. So one of my colleagues, like with him, he's a machine learning person that he's created a machine learning tool that you could actually mine the databases that are there. And that's another thing that's changed over the past 20 years. A lot more comprehensive databases and omics like platforms and tools that you can actually mine. So he's been able to mine that and predict, let's say, for microRNAs, what potential drugs can bind to microRNAs. And then you might be able to repurpose, let's say, an FDA-approved drug to target microRNAs that would be related to bad things that happen in space. But that's the one thing that definitely, like 20 years ago, would have been impossible due to the lack of knowledge that was available on the net, in general, and also the computational power wasn't there. Now those two things have evolved to a point, as we know.
So you work kind of at the intersection of two of the most cutting edge fields, where artificial intelligence and space. And I'm a big fan of science fiction, so I feel like I would be doing myself and you a disservice if I didn't ask you at least one science fiction question. So I'm curious what you think will be more important to humanity, artificial intelligence or the ability to explore space? Which one will give us a better understanding of ourselves, in your opinion? You can talk in science terms or philosophical.
Yeah, I think it's both. You could talk. I mean, I think ultimately AI is probably, to give you a better understanding of humanity, AI would probably be the step forward. And that's, you could think of anything, and that's then going to facilitate space travel, right? Because as you can look at 2001 Space Odyssey, hell, the computer, right? I mean, we're not too far away from that, right? With AI and how things evolve. But that's I think, of course, relying too much on AI at this point can also be misleading sometimes, as we know the caveats to AI. It's not perfect, right? You could. There's still a lot of kinks to figure out that way, but it provides a nice resource and tool, or what it is to actually, for example, there's a method with AI you can use called knowledge graphs. You might have heard of. And knowledge graphs uses AI to basically, depending on the question you have, it uses AI techniques to actually create nodes based on knowledge in as the name might allude to, in the internet. So let's say one aspect is, let's say if you have a question on what potential drugs can target a gene or a path or certain amounts of pathways, what it does is scour the internet from multiple pathways databases you might put in there. You might put like 50 pathway databases, and then you could also put in, let's say, a literature, like look at find the literature associated with these pathways and diseases that are related to this. And then you put in your favorite genes and drugs, and then output for drug databases. And what it does then, knowledge graph will create basically a network and a node kind of module where you have all these nodes connected based on the statistics and the AI model, and then simplifying it here, very well, simplifying it to a certain degree. And then within these nodes, then you could basically pull out the complexity of novel kind of drugs, for example, that might come out of this, or novel information and pathways that you might not have seen associated with the disease. And people are using that to actually come up and understand better diseases that, models, pathways, and genes that might pop up that otherwise manually, meaning like if we just, let's say, run our analysis on the omics data, based on what we do standard wise, you won't pull out the subtleties that we might see. And because there's no way that we could look at thousands and thousands of papers and thousands of databases that AI can do just like this, in a second. So that, in a sense, I think AI is probably the way, at least for science, it's going to be help us really push forward. Space, of course, is always fun and exciting, we said, but space itself, I mean, one thing is it's an accelerated model of aging and, as in mitochondrial disease. So space will give us an understanding of how maybe, let's say, people, I know there's probably people might criticize, why even doing research in space? We don't, so many health problems on Earth, why even bother, having this pipe dream of us colonizing another planet or us going space travel in sci-fi, as you said. And the reason, the answer to that is because, one aspect is, of course, curiosity and sci-fi, it'd be neat to do that. The other aspect is space is an accelerated model for a lot of diseases like aging and aging-related disease. So when you have, let's say, a month of experiments or even a month of a human astronaut in space, and you get the samples, and then process them and understand what's happening, you could actually then apply that back to the clinic. Because what might take, let's say, a month to do experiments in space, or what might take us a year or 2 years on Earth in the clinic to, let's say, understand muscle degeneration and diseases related to that. And space, you'll know within a month. And then you can start designing, let's say, ways of countermeasures against that, which then it could be therapeutics on Earth. So within that, AI is actually a component that you could use. So AI needs to be developed first before, well, in parallel, I should say, while we're doing space experiments.
That's interesting. I didn't really pair those together, but essentially we could learn more about human biology just by putting, maybe just mammalian biology. We don't want to go and see it, we'll put it in mice first, but we can see an abstract difference between how humans live and react to the environment on Earth and how they react in such an abstract environment with, microgravity and
right.
lack of, I guess, I don't know, probably other things like time and stuff probably would be at least a little different. And then probably you don't get like the same sun rays, and there's just probably everything is just about.
Radiation is one of the key aspects, right? Galactic cosmic rays, which are our magnetic field and ozone layer. The magnetic field reduces it on Earth, but then the ozone layer protects us even more. And then, there's a whole other, once you go into deep space, then you get fully exposed to the galactic cosmic rays and lack of gravity. But then, then there's the whole isolation, right? The isolation, which can impact your health. There's also, there's other environmental factors in space. There's say you're in a tube for a long time, right? The CO2 concentration goes up eventually. There's no way to vent it, as you can't roll down the window of the space station as you could, or in a rocket as you do in a car. So there's a bunch of other environmental factors that could impact you. And the light and dark cycle, of course, is obviously going to screw up your circadian rhythm. So, and your sleep cycle. So that's another biological impact. So there's a lot of different. Making space doesn't sound as fun as it might be, but that's why we're here to research it and make it safe and make it fun.
All right. Well, I'd like to be respectful of your time, but I do have one more question. And I did a little bit of research on you, and I found out that you make music. Am I correct on that one?
Yeah, I do. Yeah.
Yeah. So I'm also an artist. I make music as well. I do digital art, I do photography and whatever, but people always say there's a left and right part of the brain. And I feel like your work, you could say that you're, more logical and stuff, but I can see that you have endeavors outside of work that are purely abstract or artistic. And I was wondering, do they help each other at all? Does having creative pursuits help you with, more of your professional stuff where you're more involved with math and science?
I would like to claim it does. I think it does. I mean, for me, music's always been part of my life. I play in bands around town, or one band we put out original music, and we just released an album a couple of months ago. People are interested in going to Bandcamp.
There you go. I'll tag it in the description.
The band's called Medical Maps. But I think, like, for me, more music is just a way to I think everyone should, people in science work a lot because they enjoy it, but you shouldn't work all the time, because it should be a point where you can just shut off the science part and just, do something else. And this is what music is. So I get together with my friends and play music, and that kind of just shuts off the brain, part of the science part. And then when you come back to it, it helps you maybe restart in a fresh outlook, in a sense. So that I think is important, to have the balance where you could actually just kind of shut down from the science, because you could work so much, and then if you get stuck, you're stuck, right? You're not going to do it. So, and I think you're right that having that side, whether it's art or music, or having a digital landscape where you're being creative with, or writing, whatever you want to do outside of science, I think it does help creativity in a sense too. I would 100% agree. I have no proof of that, but I would agree that it does help.
That's your science side talking.
Exactly.
You're looking for proof.
Yeah, exactly. We got to do the experiments, which I haven't done. No, but I agree. I mean, I think there's a side where it does help you think a little more creatively, because you have that already, that side of you that's doing that.
Well, that concludes all of my questions. I appreciate you coming and speaking with the group. We have a couple viewers that I'd like to just give them the opportunity, if they have it, to maybe ask you a question. So if you guys in the chat have any sort of question for Dr. Beheshti, feel free. This is your chance.
Am I okay? Am I allowed to talk? I just want to make sure.
Yeah, go right ahead.
First, I just want to say thank you so much for coming. This was so interesting. And I have a million questions, but I'll try to narrow it down to just a few. First, I want to appreciate the fact that you said that when you first started doing bioinformatics, you spent a lot of time Googling like how to do things. I really appreciate you saying that, because I just finished my first bioinformatics internship, and I think half of my job was me just Googling how to do things. So it definitely is.
I still Google a lot. So I still spend half my time Googling. That's never going to stop. So go ahead.
It feels good to know it's not just me. And I guess I had a few more questions. Earlier in the interview, you said you originally worked on a kind of computational database for genes. Was it similar to BLAST?
Oh, so it wasn't specifically for, it's called Gene Lab, but I guess it's a little misleading. So Gene Lab, it has anything of omics. So it's not, it's like BLAST is a different, platform and different tool, right? That's, you want to say, a lot of people use BLAST to try to say for microbes, right? They get a sequence. What is the sequence associated with? And then be like, they use BLAST and be like, oh, it's this microbe or this virus and so on, so that's what it's used for. So Gene Lab itself is really just a repository and tool where any type of omics, whether it's genomics or transcriptomics or proteomics, metabolomics, all these different omics, any kind of big data omics gets ended up in that platform. So that would be, the data's there, the raw data's there. And then the process data, this is what Gene Lab does, which a lot of other platforms don't do, which is really great about it. They actually process the data. So if let's say you don't have the computational skill, let's say it's not like a person not like you, which is now obviously you said you're a bioinformatician, so you know how to do some of that processing stuff, but not everyone's has those skills. So if someone doesn't know how to do all the pre-processing, the process data is there. So you let's say fold change values, comparing your experimental conditions to your controls. Let's say for genes, so you get a bunch of genes and you can just look into that, so that it's basically a platform used for that. And one thing that I should have mentioned when Jeremy was asking and talking about stuff is that we also have Gene Lab early on. When I first was part of it, we established these analysis working groups, which have what is it now? I think 6 of these now exist. And these were originally established for the scientific community. Anyone can be part of this, and it can be any level of career, whether students like you guys or faculty or postdocs, graduate students, undergrads, even high school students can join, or directors, or anyone, any level of career or education is welcome to join. And Erasure is used to help develop the right pipelines and stuff so that people Gene Lab could process the data. But now we're doing fun science projects, like I lead the Multi-omics AWG analysis working group, and we have now I think around close to 300 people involved. They're all around the world, but not all 300 are active. Maybe there's like 40 or 50 people that are active, and people come and go. But within this group, we have a bunch of different projects. So right now we have about 5 papers that, we've submitted as a group. And in these papers are not the same authors. There's different authors involved, within the group. But usually these papers involve like at least 20 to 30 to 40 authors in the papers. But the great thing is, there's, some of these projects were led, for example, by he's just a first-year graduate student, and he joined the group. And before he got accepted to grad school, he joined the group. And he said, "I have an idea to work on this project." I was like, "Yeah, so it was like a good idea. Present it to the group, see who wants to help." So then that evolved to a paper. And as a first-year graduate student, he has a paper that's we're going to send back for after he answers the reviewers' questions from this paper. But he's going to get a nice paper out of it. And he's also a non-medal author and a bunch of other. But that's just an example that but that you guys, say students, want to be part of this. We meet every month in my group. Meets every month, like every third Wednesday of every month. But that's something that people want to be interested in. And if you were interested in science kind of related fun things to do and be part of projects, that's definitely more people are welcome. So sorry, side tangent to your question, but I thought I would throw that out there as a plug.
No, I actually really appreciate it. One of the reasons I chose bioinformatics is because I never wanted to stop learning. And I feel like this is a field where there's just so much to learn. And the fact that it's even applicable to space is just very fascinating. So anything you say, I'm very, very interested. The other question I had, and thank you for answering that one, was when you mentioned that you guys are doing these experiments, I had never thought of space as kind of like a advanced model of Earth kind of. But when you get that data back on the bioinformatics side, what are you guys looking for when you analyze it that would enable it to go be applied to human trials?
There's a lot. There's basically the question is to analyze this data is the limit is your creativity. So it goes back to creativity wise. The more creative you are, the more inner child you have. To maybe Jeremy's question, if you play music or arts, you could be low. But everyone has creativity once they have to just tap it. So typically for me, like since I would have a specific grant that I work on or a project, some of the omics data that I'll look at is, let's say, mitochondrial specific pathways and functions. And I have we have pipelines developed to look at specific mitochondrial and different custom pathways to look at. But the first analysis we would do in these omics data is just a general analysis. Run all these tools, like these pathway analysis tools that I'm sure you're very familiar with now. And then look at the, let's say, overall expression of how genes might be behaving. And let's say if it's a transcriptomic data. And then you can pull out, based on that, some of the key pathways that might be dominant in that data set. That might help you generate some more hypothesis to be like, oh, let's say it looks like this immune pathway is dysregulated in this organ based on the experiments that were done. So let's dive down that path and look at what's happening. And then that's one way. Then you get answer some questions there. But then there's of course 100 different ways you could diverge from that and find different biology that's changing. Now, if you have, let's say, specific questions and as I said, the mitochondrial one that I'm interested in, so I would always analyze the mitochondrial specific analysis in addition to the more general analysis. And sometimes mitochondria might not change depending on what I'm looking at, which is fine. And that's good as an answer to say, okay, there is no bioenergetic changes due to this experiment that we did and things like that. So really, the answer to your question is there's no defined way to analyze it. It's you would want to run your basic general analysis to find the up and downs, and then look at some key pathways. But then after that, it's your creativity that how you want to analyze this. So sometimes what I would do is to say on GeneLab, there's tons of different experiments on there. And one project for that analysis working group we're expanding on the mitochondrial analysis, and we're trying to analyze all the data sets that are on there and look even from plants, because there's, of course, people send plants to space and other things to space, insects, right? And insects, some insect models like Drosophila fruit flies relate to different aspects of human biology, or C. elegans of worms, and the nematodes might refer to different parts of human biology. So now we're trying to figure out how between cross species, some universal mitochondria drivers might be driving, and then that might then be applied to the human biology that changes. But that's just one example. Again, you might have 10 different ideas, 20 different ideas. I'd be like, hey, I want to start playing around and piece the puzzle together, because basically it's a puzzle, right? You have all the components that people run separate things on. Now you have to piece that whole puzzle and figure out, okay, what's the bigger picture and what's really driving human response. So then when you're in space, let's say, you're looking at mitochondrial, I'm going to focus on mitochondria again, but this is one example. Like, you say, okay, mitochondrial advances rapidly in space. These are the reasons why. Now you have some potential drugs that could target this as a countermeasure. Now you go back to the clinic. I'm a roundabout, long answer to your question. Now you go back to the clinic and say, oh, there's mitochondrial diseases in the clinic. There are a lot of them that happen in deficiencies in mitochondrial diseases. And then you could apply these same kind of drugs to the clinic if it's, to the similar disease pathologies that you see, in the advanced version in space compared to Earth.
Wow, that is so fascinating. Thank you so much for sharing. I really had no idea that like medicine could even be applied from space. You always picture like just space exploration, I guess. So that's fascinating. Thank you for sharing. And I will just ask one more question, if that's okay. For someone who would potentially want to work like in your project or on your group, like you mentioned a little earlier, what are kind of the most prominent skills you would look for in an applicant or potential person to help or student?
Yeah, so the analysis where you go, anyone can join. I'm not stringent with people coming in, as long as you're not disruptive of the group, which most people are not. No one has been, I should say, since now, because everyone's excited to be working on space. So it could be anyone has of course, if you have computational skills, then you're more valuable to the group, because then you're like, oh, I can work on many different projects. That's always needed. To process the data, to analyze the data, make figures, and then creatively kind of say how to do the analysis, But you don't have to have computational skills to be part of it. We have people who are part of the group who are more biologists, not bioinformaticians, right? Wet lab biologists. And they come in and to help, for example, on papers to help interpret the data. So let's say you came to the group and you did all the computation stuff, you got the data, you generate data, you say, oh, this is interesting, but I'm not familiar with the genes that popped up. So there might be someone who's more, in the biology, wet lab side that might be familiar with some of the genes, so they could apply their knowledge, which we have those people. Or we just need people to help, when we generate the data and write the paper, helps research the literature, because that's also another big time-consuming part, is reading the literature once we have the data, because no one's going to be expert on everything that we come up with. There's going to be all these genes that might pop up that I have no idea what these genes do, so we have to read the literature. But someone assessed with that aspect of it and becomes an expert in all the data and analysis, then that helps the writing. So there's all different aspects to people could help, from, let's say, the computational sides with methods and analysis and making figures and interpreting data, to actually helping writing. For one of the papers we included, NASA has this gene lab for high school program students that high school students go, and we even had authors that from high school, students that became authors in the paper, because that's what they exactly did. They went to the literature and they said, all right, create a table, comprehensive table of everything related with skin related damage to space. So they did a great job. They came up with this like nice comprehensive, they provided summaries of them, and that really assisted helping writing. And that helped them, that helped us out, and they were more than happy to help them out, because they became an author in the paper in high school, which is
that's incredible. Yeah. Thank you so much. What would, if I were interested in possibly attending, would you be comfortable with us on at Mesa reaching out to you?
Yeah, just email me. Yeah, you just email me and I include you guys. Yeah. And I think Jeremy has my email, or he could post.
Perfect. Thank you so much again. I really appreciate you letting us nerd out with you.
Yeah. Anytime.
Yeah, I'll be sure to include a contact link for Dr. Beheshti, because our organization is a bunch of students hungry for opportunities with computational skills. So I can't really think of a better funnel together.
I mean, unfortunately there's no funds associated. People don't get in the analysis. No one gets paid. I don't get paid. No one gets paid to do it. They're just working on it because they love it. So and eventually, hopefully, it leads to I know there's been some students on there that have been able to use that network and met, let's say, faculty, and then once they got done with their graduate school, they got hired as a postdoc by the faculty. So there's opportunity. Obviously, as I said, the network is one of the things that I encourage everyone to do in any level of your career, and this is one way of doing it. Networking is that you have fun, you work on some projects when you have time. You don't want to fail out of school, so don't join this and you fail out of school because you got too involved with this. but balance it. And then once you did that, there's ways of, obviously, opportunities will pop up in the future because they'll see what superstars you guys are, and then be like, oh yeah, of course we want to hire Jeremy out of undergrad or graduate school because or Sophia, all of that. So
I just have one question. So you said you use AI to analyze the data you receive from these experiments. Is that right?
yeah, that's one. Yeah, well, you could use it to analyze it, then you could use it to also expand on analysis too. Yeah.
Okay. So do you use AI to find relationships between data relationships that you didn't or you hadn't thought of?
Yeah, 100%. I mean, that's one thing. I brought up the example of the small drug molecule version of the machine learning tool in AI we're using. So there are some basic tools that you could, I'm sure you might be familiar with, that you can go up. There's like these tools and platforms out there that you could enter your list of genes, for example, and then say what drugs can target these genes. Just simple tools out there. But then you get a list of hundreds of drugs, and you're like, okay, how do I whittle this down to the right drugs? Right? So that's what that was my collaborator. He used that machine learning tool to actually come up with a way that it used to generate knowledge that we would have done otherwise. And it came up, let's say, for the space related work, we whittled it down to like 10 small molecule drugs that potentially could be repurposed. And then it would lead that we'd have to do experiments on and figure out and use these drugs to do it. But in the meantime, before doing these experiments, we looked at literature, and we see that a lot of these drugs that he came up with in the 10 make sense. We did the same with the COVID work. He came up with the AI technique, and luckily, well, not luckily with COVID, but the one thing, sometimes time validates your models that you do with the computation. And this is the case here where he predicted some potential drugs as targeting a microRNA that's associated with COVID. And a year and a half after he made those predictions that we published in the paper, we started seeing other papers coming out. If you use this drug, it's beneficial for COVID. If you use this drug, it was like, hey, look, after time, your models actually was pretty good. We didn't have money to run wet lab experiments at the time, but that's the limitations of science too, getting fun. But yeah, so AI ML has helped accelerate the knowledge finding process, and I would encourage everyone to use it if they can, or collaborate with someone like I do. Collaborate with a bunch of people. Yeah.
So you said your team is pretty diverse in their skill set, and I'm studying computer engineering, and I was wondering, do you have any computer engineers that are working on your team?
Yeah, I mean, I have a few collaborators who are similar to your background. So one of the projects that we worked on that is right now under review, it was focusing on how skin, space radiation and space environment might impact your skin biology. And the lead author, one of the lead authors in that paper, he's getting his PhD in computational not biology, I guess, similar to your background, not engineering, but more in the maybe computational engineering, I would say, but computational software, so he's asked it. But within that, we actually got another person who just got accepted to graduate school. He was working for a company in Denmark that was doing exactly computational engineering. So he has all these nice ML tools that help generate basically, you put in the transcriptomic data, and within the ML tool really basically came up with some key targets of genes that were going to be key hubs that might be related within that big data set. And so he's basically, like, it sounds like your background, he used his skill set to actually come up and help generate some really novel genes and pathways that we wouldn't have found otherwise within those data sets. So yeah, I mean, a long answer to your question, yes. But there's definitely use for people like you, a very good use for people like your background and your skill sets, because as you know, biologists are not as computationally inclined. So once they have someone with your skills, they tend to gravitate around you, be like, hey, can you do this? And you might get too much work.
Awesome. Thank you so much.
All right. Well, I think that concludes the other questions. I don't even know what to call it, but nevertheless, Dr. Beheshti, I really appreciate you giving us a little bit of your time. I look forward to all the publications and research papers that you come out with, and hopefully we can get some students connected with you to work on the working groups.
Yeah, no, love that. Yeah, no, thanks for having me come in. It was fun.
All right. Well, have a great rest of your day and talk to you later.
Yeah, you too. Yeah, thanks,
Thank you so
Jeremy.
much. It was so nice to meet you.
Yeah, nice meeting you. Thanks a lot. All right, bye.